Jupyter Notebook systems. And I'll explain to you what all of those are, but first I want to tell you a little bit about the lineage of both of these kinds of systems. So we're talking about two fairly different philosophies on building software and different objectives behind them. First, analysis notebook environments. These follow the literate computing philosophy, and that's a philosophy that says that you should combine your code, the actual code that you're executing, with the explanation of that code, so that everything that's necessary to understand and edit that code is all in one place. The second is that these systems contain multiple different types of data. So they use text, code, multimedia, etc., that can all be embedded into a single document. This forms the complete description of a research project, and the term that's used is the full research narrative. And the vision there is to go beyond the PDF, because there is the thought right now, which is getting more and more accepted, that the PDF, although it is not dead, it should be dead, because there is so much more that can be gained by making scientific research as live as possible. Part of that is that they can be run to reproduce the author's results and also adopted to suit the scientific requirements of researchers who may benefit directly from that research. And some typical examples here are Sage Math and, of course, Jupyter Notebook itself, but there are also examples such as MATLAB, Mathematica, countless examples that have different objectives, but all fall into this basic category of analysis environments. On the other side, another genealogy, if you will, are Bioinformatics Tool Aggregation Portals. And the objectives here are to take advanced methods that are based on code and to wrap them and make them very accessible to non-programmers. And these systems typically have additional functionality that allow you to, for example, chain multiple analyses into workflows, recording of analyses so that you can go back and see what you have previously done and rerun any analysis that you've performed. All of these software installation concerns are removed from the scientific user because whether something is in Python or C, etc., is irrelevant to the user. And there's also transparent access to compute resources because the server itself is the front end and the back end is transparent to the user. So whether you're running on a laptop or on a compute farm, the interface to these types of applications is the same. And typical examples of these are Galaxy, in Europe Mobile, and what we'll be talking more about, GenePattern. So what we did was we integrated GenePattern and Jupyter to form the GenePattern Notebook environment. So what does this get you in terms of benefits? Notebooks that are created in this environment are shareable, executable, and they are complete research narrative documents. They support in silico research from inception through publication. So what does that mean? That means that the idea of having different tools that you have to deal with when you're designing an experiment, and then as the experiment evolves, you have to get your data into shape and get it into a final format for publication. All of that can be done within this notebook environment. It is flexible and editable enough, and at the back end, it is production quality enough so that you can use the same environment for all the phases of research. It is a seamless integration of GenePattern with Jupyter Notebook, so you get the best of both types of environments, and I'll be saying more about that soon. And there is no programming required, so if you are a scientific user or if you are a computationalist who just wants to focus on perhaps the method that you're interested in and not all of the surrounding accessory methods, this provides you with a great amount of leverage. So let's look a little bit more closely into the GenePattern Notebook environment. So the idea of the notebook metaphor, is that a notebook is comprised of cells. This is the basic part of a notebook, and each cell is a different type. For example, there are code cells, and these are cells where typically the default language is in Python, but in the case of Jupyter, there are something on the order of 20 languages that are also supported. So you paste your code into these cells, and when you essentially click enter, these the code in the cell is executed and the result is returned to you. So in addition to being a scientific research platform, you could also think about this as a code integrated development environment. For annotation of your research, there are text cells, and the formatting of text is done in the language called Markdown. For those of you who are not familiar, Markdown is a simple text-based way of applying formats to text. For example, if you have 1 pound next to a line, that will cause it to be in the format of a header. And there are also easy text-based ways of making things bullet lists and tables, etc. You can also use HTML if you're familiar with that, but for the GenePattern environment, we've also gone beyond that, so I'm going to tell you about that in a second. Notebooks also provide a vehicle for reproducibility. They can be named and saved, of course, and you could also checkpoint notebooks so that you can return to the state of any given notebook at any time that you decided to save that notebook and create a checkpoint. And that looks as follows. You can basically declare that you just want to revert to a checkpoint of the notebook that you saved at this particular date. So what is GenePattern? For those who are not that familiar, GenePattern, as I mentioned, is a bioinformatics tool aggregation portal. So what does that mean? It wraps sophisticated analyses in a very easy-to-use interface. So rather than having to use code, you fill out parameters, you upload your input files, you click Run, and your jobs are run on a server, and that server can be on your laptop, or there are a number of different servers that are available. And there's a number of additional features for reproducible research. The general design of GenePattern is that there is an analysis engine that choreographs a number of different functionalities. One is a collection of modules. These are the actual analyses. There are also visualization tools that are capable of showing visual representations of any type of data that you can analyze within GenePattern. There's a pipeline environment that allows you to chain analysis modules together in such a way that they can be shared, edited, and published. And there are a number of different user interfaces on top of the module repository, pipeline environment, etc. Crucially, it is also very easy to add your own tools to GenePattern using a HTML-based module integrator. So without the need to write any additional code, you can use a local version of GenePattern and run your own tools within that as well. GenePattern is a mature tool. It was first released in 2004, and its most recent release is this past June. We have over 47,000 registered users, and the main public server that is used for GenePattern runs between 2,000 and 5,000 analyses per week. And we also have a community site where GenePattern users can contribute their analyses to the broader GenePattern analysis community. It's called GPark. And we've had contributions of analysis for CRISPR, bisulfite, flow cytometry, and many more. And this is one thing that helps us to stay at the forefront of analysis. So how does this play inside a notebook environment? Well, the first thing that you do is you log into one of those GenePattern servers that I mentioned. And that consists of inputting your user credentials, your username and password. And that provides you with access to any GenePattern server that is reachable online, such as the main server that I just described. You can also access your own local server this way. And additionally, within the same notebook, you can connect to multiple different GenePattern servers. And the number that you can connect to is unlimited. Some of the available servers include the server at the Broad Institute, which we're migrating to the Amazon cloud. There's also a public server at Indiana University, and that's of use for any of those of you who have compute-intensive requirements, because that's backed by the Mason high-performance compute cluster. You can run it on your own machinery, whatever that may be. We also provide an Amazon machine image for those people who want to run a GenePattern server in the Amazon cloud. So how do you select the analysis that you want within a notebook? The notebook provides a tab which opens to show a window that looks like this. This is a sample of the analyses that are available. You type in what you're looking for, and you can type either by name, by description, or by category. So, for example, you can type in gene expression, and you'll be presented with all of the gene expression modules available on the GenePattern server that you're connected to. What's available on the main GenePattern server? There are over 200 different genomic analyses that you can select from. These include gene expression analysis, sequence variation, SNP and copy number, network analysis and reconstruction, proteomics for MS and CellD and MALDI technologies, flow cytometry, as well as general machine learning methods, including your typical clustering, classification, and dimension reduction. So basically, most of what's available in functional genomics you can get through GenePattern, as well as the crucial utilities that are sort of the mortar that formats files and all of the in-between steps that you need to actually go from one step to another. So once you've selected an analysis, you see an interface that looks very similar to the interface that I showed you for the public genome, the GenePattern server, where you're provided with the option to input files and select parameters. And once you've done that, you submit the job, and that runs on a GenePattern server. You're then presented with a GenePattern notebook job cell, and that informs you of the status of your running job, which is pending, running, or completed. And it also presents you with a display of your result files. So there are a number of different things that you can do with result files that show the seamless interaction between GenePattern and the notebook environment. And I'll show you that in 1 second, but I also wanted to show you the additional cell type that we've created for the notebook, which is the rich text editor cell. This allows you to add text using the Microsoft Word type of interface. So you can format your data basically the same way that you would do in Word without having to worry about HTML or any type of markup. So how does this play with Python? For those of you who are programmers and computational biologists, we have created an environment where it's not just a situation where you can either do Python or you can do GenePattern. You can flow your Python into GenePattern analysis cells and out of GenePattern analysis cells into Python again. So how do you do that? So for example, if I want to, instead of uploading a file as an input, use what's in a Python variable as an input, I can, for example, declare a Python variable here, and this says preprocess floor equals 100. It's for the people in the back who can't read it. So now, instead of having to actually use a number here for this parameter floor, I am now using my Python variable, and I just annotate that by 2 braces on either side. And so I've used something that is in Python within the actual GenePattern analysis. So on the flip side, after GenePattern is finished creating a result file, that exists as a file somewhere on a server. If I want to get that into Python, how do I do that? I can click on this eye icon here, and that presents me with a number of different choices. I can download the file, I can open it, I can send it to code that will send it to a Python variable as an unstructured, basically string object, or I can send it to a data frame. For those of you who use array type data, this is a very, very quick shortcut to do that. After I click send to data frame, I see that another cell has been created for me that contains all the necessary code to import the contents of that file, and I now view the contents of that file as a data frame. And those of you who are Python programmers who are familiar with the data frame object know that there are a tremendous number of ways that you can manipulate data once it's in the data frame. So I just want to take a step back and highlight what has just been done, because it really emphasizes what the notebook integration with GenePattern is good for. So I was talking about clustering methods and genome analysis. If you wanted to do these things in a notebook without GenePattern, you would need to do the Python programming yourself. So you would need to identify a Python package, let's say scikit-learn, that had the necessary analysis that you wanted, let's say support vector machines, you would need to then import that package, write all the surrounding code to import the input data, run the analysis, and export the output data. So even though Python provides you with a tremendous amount of convenience in terms of all those functionalities that I just said, that will still take you on the order of 100 lines of code. So what we've done is to create a cell type where instead of running potentially 100 or more lines of code, you insert one cell and that does the job that that code would have done. So how can you use GenePattern Notebook? We have provided an online repository that you can go to, and accounts on this are free, and it runs in the Amazon cloud. And when you sign up, you get your own account and you are presented with a number of different notebooks. These notebooks get you started with both the mechanics of GenePattern Notebook, and also they present a number of different analysis scenarios that may be of use in your own work. For example, a general differential expression analysis for microarray data, and we have a separate notebook for RNA-seq differential expression analysis. And we have a number of different notebooks for those general types of machine learning issues. You can also contribute your notebooks to the public simply by declaring your notebooks public. And when you do that, others are able to find your notebooks and incorporate them into their own GenePattern Notebook repository accounts. So there are a couple of important points behind what we've created. The first one is that this is, to our knowledge, the only place where you can go and do GenePattern and Jupyter Notebook work online without charge, where your notebooks can persist. We know that there are tools where you can create your own notebooks, such as tempnb.org, where you can do some work, but then as soon as your session is over, that notebook disappears. And there are other popular tools that let you view the output of a notebook and persist that output, but you can't actually dynamically interact with that notebook. Using this resource at genepatternnotebook.org, you can do both of those things: create, compute with, and develop notebooks, and you can also disseminate your notebooks to the public using the same resource. So what if you wanted to use this yourself, or you didn't want to use the online version? For some reason you want your own copy. There are two ways that you can do this. The first is that we make the entire online repository with all of the requirements available as a Docker file, and you can go to Docker Hub and download GenePattern Notebook and just get started with any Docker container system that you can have. Also, for those of you who are Python programmers, we make this available as a pip or conda install. And so finally, there is a paper forthcoming in Cell Systems that has more information. So I encourage you to go there or to the GenePattern Notebook site. And I'd like to acknowledge the GenePattern development team, as well as our collaborators on the Jupyter Notebook team. And I'd like to take any questions.
Thank you very much, Michael. Do we have any questions? Would you like to talk on the mic? Yeah.
Well, if I want to use my own notebooks in it, how do I use the dependencies? Because can I also use conda to inside my notebook? So I want a separate notebook and a conda install for my notebook. Can I do that?
So the question is, if you want to use your own dependencies, can you do that? So the first answer is the repository comes already with a complete Anaconda distribution attached to it. So hopefully there should be very little dependencies that you need that aren't already there. However, if you have dependencies on a different package version, right now we are working on a strategy that allows for package version management. And there is a system right now that exists for that. I'm forgetting the name of it, but we're starting to incorporate that. So you will be able to specify with a notebook all of the package dependencies and also their versions along with that. So that'll travel with each notebook. Okay. So the short answer is yes, soon.
Okay, thank you.
Okay, we've got one more question.
Thank you for the nice talk. I have a question regarding some packages which require other packages to be in a certain version. Do you take care of that or not?
So yes, so the answer is very similar to the previous answer. We have by default the most recent Anaconda package versions, and we are working to incorporate a Conda package version manager, if you contact me, I'll have the name of it, but it's just escaping me right now. But there is a package manager that we're currently integrating into the repository. We recognize that it's really important that people be able to import the versions of the packages that are compatible with their notebooks. So that's going to be essentially in our next development sprint.
Okay. Thank you.
Okay, well, thank you very much for those questions. Thank you
you.
very much, Michael.